Students, today we will study chemistry of nucleic acids. As you all know, nucleic acids are macromolecules which are present in all living cells in combination with proteins to form nucleoproteins. The proteins present in these nucleic acids are usually protamines and histones. So all the genetic information is encoded in a nucleic acid molecule, and nucleic acids means mainly they are of two types: DNA deoxyribonucleic acid and RNA ribonucleic acid. And these nucleic acids, they are nothing but polymers of nucleotides which are linked to each other by phosphodiester bond. That's why they are also called as polynucleotides. And we all know DNA is present in the nucleus of the cell. That is different. Small amount of DNA is also present in the mitochondria. So the DNA, which is mainly involved for the formation of all the proteins in the body, is mainly present in the nucleus of the cell. So first we will study what you mean by the term nucleotide. So each nucleotide is composed of three components. They contain a nitrogenous base, a pentose sugar, and a phosphate group. And in nitrogenous base, again, it can be of two types. It can be a purine basis or pyrimidine basis. These both purines and pyrimidines are present in RNA and DNA. So two main principal purine bases in DNA as well as RNA are adenine and guanine. Say this is the structure of the basic purine ring, and adenine is nothing but 6 amino purine, and guanine, it is nothing but 2 amino 6 oxo purine. So this is the basic structure of purine and pyrimidine ring. This is only for your understanding. You need not practice or by heart any of these structures. These structures are only for your understanding. So coming to the pyrimidine bases, three major pyrimidine bases which are present in DNA and RNA are cytosine, uracil and thymine. Cytosine is the pyrimidine base which is present in both DNA and RNA, whereas thymine, it is present only in the DNA. Uracil, it is present in the RNA. So cytosine and uracil are the bases which are present in the RNA, and cytosine and thymine, they are present in the DNA. Both DNA and RNA contain the pyrimidine cytosine, but they differ in their second pyrimidine base. That is, DNA contains thymine, whereas RNA contains uracil. So again, this is a structure of the basic pyrimidine ring, and cytosine is nothing but 2-oxo-4-amino pyrimidine. Uracil, it is nothing but 2,4-dioxypyrimidine. Thymine, it is nothing but 2,4-dioxy-5-methyl pyrimidine. I am again telling, there is no need to by heart or practice these structures. The structures are only for your understanding. Next, we will come to the pentose sugar, which is present in DNA and RNA. And we know the pentose sugar, it can be either D-ribose or D2-deoxyribose. DNA contains deoxyribose and RNA contains ribose. And the pentose sugar is linked to the base, that is nitrogenous base, which is a purine or pyrimidine, via N-glycosidic bond. and one term is there nucleoside. Nucleoside, when the nitrogenous base combines with the sugar, it forms a nucleoside, and if you add a phosphate group to this, it becomes a nucleotide. So this is the structure of ribose, and this is the structure of deoxyribose. The only difference between this structure is, if you see at the second position, there is OH group present in ribose, and here the O is removed, only H is present. And the carbon atoms present in the pentose sugar, they are named as 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime. So the sugar present in RNA is ribose, and the sugar present in DNA is deoxyribose. And in this you can see the N glycosidic bond. So if you see, between the first 1 prime carbon of ribose is linked to N9 of purine ring. Otherwise, the first carbon of deoxyribose, it is linked to first nitrogen of the pyrimidine ring. So if you add one phosphate group to this, they become nucleotide. If only nitrogenous base and sugar is present, means it is a nucleoside. That is the only difference between nucleoside and nucleotide. So the nucleosides of adenine, guanine, cytosine, thymine, and uracil, they are named as adenosine, guanosine, cytidine, thymidine, and uridine respectively. So adenine becomes adenosine, guanine becomes guanosine, cytosine becomes cytidine, thymine becomes thymidine, uracil, it becomes uridine. So if you observe here, the purines, the nucleosides of purines, they end with sine, S-I-N-E, and the nucleosides of pyrimidines, they end with D-I-N-E. So if the sugar is ribose, ribonucleoside is produced. If the sugar is deoxyribose, a deoxyribonucleoside is produced. So next we will study the structure of this nucleotide. I have told you nucleotides are nothing but phosphorylated nucleosides. Means nucleosides contain a nitrogenous base and pentose sugar, and nucleotides, one more phosphate group is added. That's all. And depending on the number of phosphate groups, nucleotides, they are called as mononucleotide, dinucleotide, or trinucleotide. And nucleotides, they are of two types. One is deoxyribonucleotide and one is ribonucleotide. The only difference between this deoxyribonucleotide and ribonucleotide is the presence of the pentose sugar. That is, deoxyribose is present in deoxyribonucleotide and ribose is present in ribonucleotide. So this is structure of nucleotide. Already we have seen this. The nucleotide, it contains a nitrogenous base. It can be a purine or it can be a pyrimidine attached to a pentose sugar. The sugar can be ribose or deoxyribose. And to this, if one more phosphate group is attached, that forms a nucleotide. So mononucleotide means only one phosphate group is there. So mononucleotides are nucleosides in which a single phosphate group is attached to the OH group of the pentose sugar. See example, adenosine monophosphate. That means it contains the nitrogenous base adenine, pentose sugar ribose, and phosphate. If an additional phosphate is added to this, so AMP gets converted to ADP, adenosine diphosphate. Then one more phosphate group if you add, it becomes adenosine triphosphate. So depending on the number of the phosphate groups, it can be monophosphate, diphosphate, or triphosphate. See here structure. So adenine, it is a nitrogenous base. You add a sugar, it becomes a adenosine, which is a nucleoside. And to this, if one phosphate group is added, it becomes a nucleotide. As there is only one phosphate group, it's called adenosine monophosphate. Two phosphate groups, adenosine diphosphate. Three phosphate group, adenosine triphosphate. We all know ATP or adenosine triphosphate. It is the energy currency of the cell, and it is one of the most important nucleotide. So next we will come to this question, biologically important nucleotides. We know that this DNA and RNA, they are nothing but polymer of nucleotides. So the basic function of nucleotides is they are involved in the formation of this DNA and RNA. Other than that, there are many nucleotides which are having this biological importance. So besides being the structural component of the nucleic acids, several nucleotides like ATP adenosine triphosphate, ADP adenosine diphosphate, cyclic AMP, GTP, GDP, cyclic GMP, UDP, CTP, CDP, they participate in several biochemical and physiological functions. Such nucleotides are called as biologically important nucleotides. That means the nucleotides which are not the structural components of the nucleic acids, but they perform still several biochemical and physiological functions in the body. They are called as biologically important nucleotides. So we will study one by one the biological important nucleotides, and of those adenosine triphosphate is the most important. We know ATP, it serves as the energy source in the cell. For all the metabolic processes, ATP, it supplies the energy. So whatever the metabolic pathway it may be, glycolysis, TCA cycle, cholesterol synthesis, protein synthesis, energy is required, and energy is supplied in the form of ATP. Other than that, the physiological functions like muscle contraction, nerve impulse transmission, functioning of the kidneys, or our heartbeat, all these require energy. So energy is supplied in the form of ATP. Next is AMP. AMP adenosine monophosphate. It is a component of many coenzymes such as NAD, NADP, FAD, coenzyme A. See, NAD means nicotinamide adenine dinucleotide. NAD, it is a coenzyme form of the vitamin B3, that is niacin. FAD, it is a coenzyme form of riboflavin. The main function of coenzymes is they are involved in the metabolism of all the macromolecules like carbohydrates, proteins and lipids. They are very essential for the metabolism because the coenzymes help in metabolizing the carbohydrates, lipids and proteins. So this AMP, it is a component of many coenzymes like NAD and FAD. Next, number three, cyclic AMP. Cyclic AMP is formed from ATP by the action of adenylate cyclase. See, this adenylate cyclase enzyme is present in the cell membrane. So hormones, they act as first messenger. So this cyclic AMP is the second messenger for many hormones like epinephrine, glucagon. So cyclic AMP, whatever the functions needed to be done inside the cell, they are done by cyclic AMP, which is a second messenger. Cyclic AMP is formed from ATP by the action of adenylate cyclase. So it enhances the degradation of storage fuels like fat and glycogen by stimulating lipolysis and glycogenolysis. It inhibits the aggregation of blood platelets. Cyclic AMP even increases the secretion of acid in the gastric mucosa. These are the other functions of cyclic AMP other than acting as a second messenger. See, this is a long list of hormones which use cyclic AMP as a second messenger. See this calcitonin, chorionic gonadotropin, corticotropin, epinephrine, FSH, glucagon, LH, melanocyte stimulating hormone, norepinephrine, parathyroid, thyroid stimulating hormone, vasopressin. All these utilize cyclic AMP as a second messenger, and cyclic AMP, it is a second messenger and it is a nucleotide. So that is the reason it is a biologically important nucleotide. Next, GDP and GTP. GDP, you know, guanosine diphosphate. GTP, guanosine triphosphate. Like ATP, GTP is also a source of energy, and GTP, it is formed in one reaction of the TCA cycle where succinyl CoA gets converted to succinate. It is known as substrate level phosphorylation. GTP is required for the activation of adenylate cyclase by some hormones. GTP, like ATP, also serves as a source of energy for protein biosynthesis. After this, cyclic GMP, like cyclic AMP, cyclic GMP is also a second messenger, and cyclic GMP is formed from GTP by guanyl cyclase enzyme, and cyclic AMP, GMP, is involved in the relaxation of smooth muscles and vasodilatation. Next, biologically important nucleotide is UDP, uridine diphosphate. Uridine participates in glycogenesis. See this UDP glucose. From UDP glucose, this glycogen can be synthesized. And UDP galactose, it takes part in this galactose metabolism and it is required for synthesis of lactose and cerebrosides. Other than this, UDP glucuronic acid is required for detoxification process and for the biosynthesis of mucopolysaccharides such as heparin and hyaluronic acid. So here UDP, uridine diphosphate, either UDP glucose, UDP galactose, UDP glucuronic acid, these all are biologically important nucleotides which are involved in the different metabolisms. Next is the CTP and CDP. Both are required for the biosynthesis of phospholipids. Like CDP choline, which is involved in the synthesis of sphingomyelin. So other than this, one term is there, they are called as synthetic analogues of nucleotides, also called as anti-metabolites. So an analog is prepared either by altering the heterocyclic ring or the sugar moiety in the nucleotide, and these are used mainly in the treatment of cancer or even HIV infection or any viral infections for that matter. So see this 5-fluorouracil, 6-mercaptopurine, 6-azaruridine, these all come under the group of this synthetic analogs. So analogs of purine and pyrimidines which are used in the treatment of infection or in cancer chemotherapy. Other than that, cytarabine, allopurinol, which is used in the treatment of hyperuricemia and gout, it is also nothing but a synthetic nucleotide. Other than that, azidothymidan, which is used in the treatment of AIDS, it is also nothing but a biologically important nucleotide, or you can call synthetic nucleotide. Other than this, this azathioprine, which is catabolized to 6-mercaptopurine, it is also used as a immunosuppressive agent during organ transplant. And 5-iododeoxyuridine, a nucleoside analog, has antiviral activities and it is used in the treatment of herpes infection caused by herpes virus. Okay.